Our next panel is going to be a similar panel, but with a discussion really focused on molecular and genomic profiling and targeted therapies. So I'm inviting Eric Connick and Anna Berry to join me on the panel, and I will let you introduce yourselves. Then I'm going to give a brief introduction to the topic, and then we'll go back and do a panel and with questions and answers. So, Eric?
Hello. So I'm a... I'm Eric Connick. I'm one of the medical directors at the University of Washington.
It is on, so I see the green light, so hold it a little closer to you.
Okay, so I'm Eric Connick. I'm one of the medical directors at the Genetics and Solid Tumor Laboratory at the University of Washington. So we do clinical testing. So this is testing for patients who are being seen right now for treatment reasons. So we'll test tumors for different markers. And then we also do germline testing, so looking for things like BRCA mutations. And we do that in collaboration with Mary Claire King. And there's a group of 8 of us who collaboratively do this for direct patient care.
Hi, I'm Anna Berry. I'm also a pathologist. I do molecular pathology and cytopathology, so like the FNAs. I work at the Swedish Cancer Institute. I'm the scientific director for the personalized medicine program there. I also am working at Cellnetics, which is the pathology group that works at Swedish. So we together designed a targeted cancer panel that is the core of the personalized medicine program. So we're really looking for targeted therapies in a systemic way. I think Dr. Brown might talk a little bit more about the details of our program a little later. But I worked at UCSF for almost 10 years doing molecular oncology there, so I have spent a lot of time devoted to trying to find molecular targets and alternative therapies. Ironically, the reason I don't look like my picture up there is I also am a breast cancer survivor. The past year, I was diagnosed with breast cancer and had the joy of undergoing Cytoxan Taxotere therapy. So, I understand it a bit even more now, how important it is to find some alternatives.
So, great. Another patient scientist
Right.
Today. So I'm going to give just a little background about why we care. We've got these experts up here on molecular and genomic profiling, and why we care about this. So we know that cancer is actually a disease of genes, and over our lifespan we have a lot of exposures that cause genetic changes that take a normal cell to a cancer cell. So we can inherit some of those abnormalities, and in breast cancer we know about BRCA1 and 2, which a minority of breast cancer patients have associated an abnormality in that, but if you do have an abnormal BRCA1 or 2 protein, then that leads to a high risk for the development of breast and ovarian cancer. But we have, for the most part, external exposures, internal exposures, lifestyle factors that add up, causing multiple mutations in cells over our lifespan that can result in cancer. So all cancers are caused by genetic changes. And we've now entered the genomic era of cancer treatment, where we're better trying to understand the genetic changes in each individual cancer, what takes a normal cell and turns it into a hyperplastic or atypical cell, and then what takes it to a pre-invasive cancer, and then an invasive cancer, and then a metastatic cancer. Different genetic changes are occurring along the way, and a better understanding of those changes as they've occurred in each individual cancer can lead to better treatment strategies. So we saw a similar slide earlier this morning talking about how now our tendency is to think of breast cancer as at least 4 major genomic subtypes. Dr. Specht referred to the basal subtype, which we associate with triple-negative breast cancer, but it's not 100% overlap. We have the luminal A and the luminal B subtypes, which are more the ER positives, and the HER2 subtype. This is a heat map. So this is one way of looking at genomic profiling, and across the bottom we have a bunch of different patients, and then going up the y-axis we've got a bunch of different tumors that are being tested for in a bunch of different patients. These are all breast cancer patients and genes associated with breast cancer. And the red are the places where the genes are acting abnormally compared to normal cells. In the green, they're more the same as normal cells. And you can see here the 4 subtypes as pointed out in this heat map, where different sets of genes tend to collect as being abnormal in the different subtypes. So this breaks up breast cancer into at least 4 different subtypes. And then Dr. Specht, in talking about triple-negative breast cancer, showed you this data out of Vanderbilt that suggests that triple-negative breast cancer in and of itself is at least 6 different subtypes. So the fact is, the harder you look, the more you look at the genes and their associated proteins and how they're regulated, the more each breast cancer is unique and individual. And, as was alluded to in a question we had in the last session, we know that you have your metastatic breast cancer now, and we profile that, and then you get a treatment, and then another treatment, and another treatment. The genomics of that cancer evolves, because in order to survive and grow through each treatment, you might kill 95, 99% of the cells, but some group of cells figures out how to change really at a genomic and a molecular level and grow through that treatment. So the tumor may get smaller and then start growing back, and that's a new subset. And there's something different about that subset that's now growing. So we do need to look at genomics across the treatment span of metastatic breast cancer. Now this slide might look overwhelming, but it's actually so incomplete in terms of the number of different targets and the number of different classes of drugs that we're investigating right now in breast cancer. How are we going to study all these targets that we're finding from genomic and molecular profiling? How are we going to study all the drugs and the classes and get them, frankly, approved so patients can get access to them? We now clinically can do genomic profiling of cancers, and this shows all the different genes. You're not supposed to be able to read it, and then a potential report that would come out of genomic profiling. And we've got experts here who are doing genomic profiling and help us evaluate the amounts of data that come out of this are incredible. When we started a metastatic breast cancer project where we wanted to look at the DNA, RNA, protein level, etc., the guys who were running this project from the research end, the genomic molecular end, said, we actually, at the University of Washington and the Fred Hutch, don't have enough server space to keep all of the information we're going to get out of all these tumors. I mean, massive, massive amounts of data that we can collect, and then we have to have people who help us figure out how to sort that all out. All the informatics people who help us take these massive amounts of data from each tumor and then help us process what's important. So we have had some trials looking at doing genomic profiling and matching to drugs. This was a French trial called the SAPPHIRE-1 trial that ran genomics and then tried to put patients on targeted drugs. They looked at 423 different patients who enrolled. 46% had what they called an actionable mutation, meaning there was some drug out there somewhere, not yet FDA approved or FDA approved, that could target the things that they were finding in these patients. But based on the number of drugs they had available and the number of trials, only 13% had alterations that let them get a drug. So a big problem here is they found some mutations but couldn't get access to a drug, and then only 4 of those had a response out of 423 that they started with. The majority who couldn't get access to a drug that related to their mutation. So while this is viewed as a negative trial, it is actually a really important trial because it shows feasibility, but it also shows the problems was that we need access to the drugs that target these mutations. So we now have a big national ongoing NCI-sponsored trial called the MATCH trial, the Molecular Analysis for Therapy Choice. This is for all metastatic solid tumors and lymphomas. You have to have had treatment with at least one prior regimen first, and then you can enroll. We need to get your tumor, and then they do genomic profiling. They find the abnormalities, and then they have now, at this point, access to 24 different pathways, drugs in 24 pathways. And so if you have a mutation for which they have a drug, you can get access to that drug. You can stay on it until the tumor grows again. And then if you have a different mutation, you can get access to a next drug. So they're hoping to screen 5,000 patients, but they estimate that only 23% with the drugs we now have available will have a matched target. So we still have an issue of having drugs in these different pathways. But this is an active ongoing trial right now that some of you could be eligible for. So I'll end and go to my panel now, that this is what precision medicine, personalized medicine, whatever you want to call it, is. Over here on the left, you have breast cancer, you get this drug. That's all we know. We assign it based on just, we know this drug works in metastatic breast cancer. But over on the right, with precision medicine, we do DNA tests, protein tests, etc., and we assign therapy based on those. And while we do know about estrogen receptor and HER2, and you heard talks about this morning, we're talking about going way beyond that and looking at all kinds of pathways and taking triple negative and calling it what it's positive for. So that's what precision medicine is. And I'll end with that and now actually open it up to my panel. So why don't you both spend a few minutes talking about your thoughts about how we're going to take genomics and precision personalized medicine and really use it in patients. What's going on now and what's going to happen within the next few years? Anna?
I'll start since I have a microphone. So I would say that this is a complicated area in large part because our technology has kind of outpaced our capacity really for doing clinical research. So, we haven't really gone into the details of what we're able to do with genomics, but to summarize it quickly, in the past, we used to be able to look at one little segment of one little gene at a time using something called Sanger sequencing, and it wasn't very sensitive, but it still was like a really revolutionary technique. In fact, that's how we sequenced the genome for the Human Genome Project. Now we can look at millions of fragments in any part of the genome we want, all at once. So it's dramatically less expensive, it's faster, it's more accurate. We can look all the way down to like 1 or 2% of the material in the DNA, which is really, really important when you're looking at a solid tumor because it's never entirely tumor. These things are invasive and go through our bodies and they have inflammatory cells and stromal cells and all kinds of contaminants. So we're able to look with much more sensitivity at a huge number of targets. So because of that, we're generating vast amounts of information. We mentioned, the computing capacity, the bioinformatics, everything that's needed because it's a huge amount of information. So given that, clinical trials, as most of you probably know, take a while to put into place and get set up and get all the staff and get the tissue collection and all those kind of things. So that system isn't really adequate anymore. So drug companies now have basket trials. They have trials that are directed towards any tumor type, towards any genetic mutation. Now we have these national trials like the MATCH. We also now have TAPER from ASCO, which will soon be available across the country, where you don't need a new biopsy. You can use your existing tissue biopsy and your existing genomic test. Any CLIA-certified lab can provide information for TAPER. And the drug companies will give you the drug, right? So you can actually get access to the drug. The downside is you don't really know how it's going to work yet. All of these are new and, to some, oncologists who are very disciplined people and who like to act based on solid evidence before they give you a drug that, may affect you in strange ways. They're not necessarily, into this just randomly assigning drugs. So trials like TAPER, programs like we're doing at the Swedish Cancer Institute are trying to control that process. We're trying to see if we give you that drug and it works, can we use that for the next person? Because what we don't want is that information being out there and you get a targeted therapy and lots of people are getting that and, we should know the outcome, but we don't because it's just random. It's the person who managed to get the drug through their insurance and, and that experience is lost. So we're trying to capture all of the experience. The other thing I want to say is, we saw a little bit of discouraging evidence, right, that we don't know what targets are best when we're looking at CDK4 inhibitors or mTOR inhibitors, that very few people have a response or get access. But part of what we need to look about are those very complex pathways. Everybody would love for this to be a Gleevec, which is the drug that people got for chronic leukemia, and it becomes a chronic disease, and you take that drug the rest of your life and you're fine. Unfortunately, breast cancer is a lot more genetically complex than chronic leukemia is. There are multiple genes involved, and the genes that are involved have multiple inputs. That CDK4 inhibitor is involved in so many different pathways. So if we're looking at multiple markers, if we're looking at larger panels and we're putting more information in, we'll be better able to understand that pathway. And ultimately, we'll be able to narrow down who's really going to respond to that drug. It's not going to be just one hit that it's CDK4. The most common mutation for that is actually this mediator called CDKN2A, which is a tumor suppressor. So once you don't have that gene anymore, then you're responsive to that drug. So, clearly that can't be the only thing, and that's probably why it's not completely predictive. But basically what we're trying to do is get more information to narrow down the focus. So I guess the good news is that all of these things are very promising, and the field is really rapidly moving. I think the bad news is we're just not there yet. I don't think we're anywhere close to there. There are a few things that are effective and a few mediators that we know about. Those would be kind of standard of care testing, and I think that that's growing. The little panel that will be appropriate for breast cancer is growing, but eventually it will be huge. It will be much bigger, and all of the information we're gathering now will be relevant eventually.
Great. Eric?
All right, so I think Dr. Berry kind of covered the waterfront in terms of clinical trials and things like that, so I'll take a little bit different perspective. So I'm a pathologist and a laboratory medicine physician. And so one of our roles in the laboratory is looking at what we have available in the lab right now to benefit patients, what is available coming down the pike, and then how we can use new technologies, new insights, new ways of thinking about things to benefit not just patients in the future through clinical trials, but patients right now. Like, how can we make a difference right now? And so some of the approaches we've taken at the University of Washington is basically looking ahead what the newest, latest, greatest technologies are, and then applying those to the problems we have. There's no reason that we need to be using techniques that are 30 years old for diagnostic purposes when we can use something that's much more informative, much more sensitive, gives us a lot more information. And so that's what we've been doing. And so looking at the diagnostic realm, we see that we're getting smaller and smaller biopsies. We're getting patients who've been treated before. There's less tumor to deal with in the laboratory. So we're looking at new techniques. So one of the things that's been really hyped up in the news is cell-free DNA. And the problem is that this is just random pieces of tumor DNA that are circulating around in individuals who have cancer. And there's been a lot of money poured into it. There's a lot of hype, but we really don't understand how that relates to the biology of the tumor. And I think that's what we're going to learn in settings like the university, where we can do very careful approaches and benefit patients now as well as looking towards the future. And like Dr. Gotti said, one of the things that we've noticed in our practice is, so again, we're testing patients on a clinical basis for their needs right now, but in that process of doing that, we make observations, and we're testing all these different tumors. Every single tumor is different, but there's some commonalities between them. And so within our group, in other cancer types, we've made observations that are relatively rare, but because we're able to see these patients, across the spectrum of disease, we say there's something interesting about that. We work with our oncologist colleagues, our basic scientist colleagues, and then do clinical trials, which eventually is going to come back and benefit the patient. So, it's an indirect research type activity, but it's something that, as pathologists, we're trained to make these observations and be very careful in what we're seeing, and note those and apply those to patients. So,
Great, Erica. You brought up the cell-free DNA, and there's a question about what we call liquid biopsies, which I don't know if you've heard that term, but it would basically be just drawing blood and finding evidence of the cancer in the blood, whether it's an attacked cell or DNA floating around, that cell-free DNA, just tumor DNA floating around, or RNA sometimes. as well. And so somebody is asking about how well do these liquid biopsies correlate with what's actively going on as the tumor is progressing?
That's an excellent question, and in reality, we don't know for most cancers. So the one that we have the most experience with is prostate cancer. And prostate cancer, a tumor biopsy and the circulating cell-free DNA in metastatic patients matches almost perfectly. So those two work really well. Things like lung cancer, it's just hard to detect. Breast cancer, I don't think there's enough data to really know how well those things correlate. But it's an advancing piece of the field, but we need to do careful studies. But it's not something that should be used in routine practice at this point. Is our opinion.
Great. Somebody else asked.
Can I
Yes, go
make a
ahead.
comment regarding that? There is one commercial company that's offering an NGS test for liquid biopsies, and those results are acceptable for the TAPER trial. So I just wanted to be sure to make that point. There is some debate in the field about how valid those responses are, but I think genes that you see in a liquid biopsy, if you see a PI3K mutation in a liquid biopsy, it should be there in the tumor. There's no reason why you should have that in your circulating DNA in a very tiny amount if it's not in your tumor. And those results are acceptable for the TAPER trial.
But making the clear point, what we're studying and what we think is worth investigating and what's acceptable in a trial is still under investigation. So we don't want to leap forward too fast, but it's a really exciting area because sticking a needle in somebody's liver multiple times over the course of metastatic treatment is not a lot of fun and causes complications. Somebody asks, how long are tissues stored?
So there are guidelines, and so generally in most practices, pathology practices, it's around 10 years or so. Academic centers tend to be a little bit more conservative, and so we actually have tissues going back decades here and other institutions as well.
Yeah, I think there's a difference between the fresh tissue and the tissue blocks. So Eric is talking about tissue blocks, and they're in paraffin, they're like filed away in drawers, and they can be kept for a very long time. The fresh tissue or frozen tissue that we're talking about putting in tissue banks is a different thing. Tissue is not routinely frozen. So that's something that you would need to provide consent for up front to have your tissue frozen. The good news is we're finding out how to do lots of things with those paraffin blocks. It used to be everyone thought you needed frozen tissue, but that's not so much true anymore. The testing that we do in our labs is done on paraffin blocks.
Great. There was a question, actually, that came up with the last panel, but I held it for this one because I thought it was more appropriate. And they want to know about what's the connection with academia and what we're doing and 23andMe. So so not everybody knows what 23andMe is, so maybe a little, what is 23andMe, and then how does this relate to all this genomic profiling we're talking about?
I'll answer that one. So 23andMe is direct-to-consumer genetic testing, and the testing that I believe they're doing now is really more related just to ancestry, because they got into a little bit of trouble with the FDA about them thinking that they should be providing medical information to people without medical supervision. They also were not a CLIA certified lab, and I'm not sure what they're doing about that now. But the basic issue is, should anyone be able to just, write into a lab and get their genetic information? And, again, that's very controversial. Personally, I don't see a big problem with it. To me, we're so much more informed now than we used to be, and I don't see any problem with more information for patients. There also has been a recent change in HIPAA where you're allowed access to your pathology report, and newsflash, you're actually allowed to talk to your pathologist, which, you would think that would be, a given, right? But you could do that because we are medical doctors, and, you do know a lot about what's going on with your testing for sure. I don't see a problem with it. The problem with 23andMe to me is, they're not doing anything that's going to be useful for this crowd. I think, people might have a misconception that, you're going to do that and you're going to find out about your cancer risk genes, or you're going to find out about something that's, important for your therapy. And that's not the case. So I think, you do need to know exactly what you're getting when you sign up for those things. I don't have a political opinion about 23andMe. If you ask me about Theranos, that's another thing, like, we don't know Theranos.
So in terms of you know, direct-to-consumer testing and things like that, I think it's very powerful for us to be able to get that information and have some insight into, you know, our genetics and our ancestry and things like that. So kind of echoing what Anna said, so we've had a couple of examples where prior to the most recent version of 23andMe, where they can't report things like BRCA status, where individuals were tested and told that they were negative. And you have to really burrow down to the fine print, and it said, well, they only test for 3 mutations. And they're the 3 most common mutations in Ashkenazi Jewish individuals. And, you know, that's great, but, you know, we've had, again, multiple cases where individuals came into medical genetics because of their family history, said, oh, I'm negative by 23andMe, and then we do the testing in our lab, and, you know, lo and behold, they have a very severe mutation that predisposes them to cancer risk. So, you know, you have to take everything with a grain of salt, and, you know, again, very powerful, but for medical indications at this point, that's not the use of those particular services.
Yeah, and I think, you know, just that is the perfect example. I've had patients who, by 23andMe, think they've had genetic testing, and it says, you are at low risk for breast cancer, and they're not at all. And they end up, because, you know, they just don't have so it's all about the interpretation. That's the thing. I have no issue with everybody having access to everything we know about the genes, but it has to be done in the context of having the interpretation with it, and that is part of the problem with 23andMe, for example, and some of these others, is you need somebody to help you interpret the information that comes out, and we don't know how to interpret everything yet. So, yeah, I'm told that I'm supposed to wrap up the session. Did you want to add one?
Oh, no, I was just going to reinforce that, you know, really, the interpretation is what rubber meets the road, and having experts, you know, not only in the disease, but in the gene and the pathology is really key. And if that piece of this type of testing on a clinical basis isn't always there. And so, you know, the tests of the different laboratories are different, but it really comes down to the experts, you know, behind the microscope, behind the sequencers, behind the